Hello everyone, welcome back to my channel. Today we'll start with a new topic that is known as fluorescence spectroscopy. Fluorescence is a physical phenomena where certain substances called fluorophores or fluorescent molecules absorb photons at one wavelength, that is the excitation wavelength, and then re-emit photons at a longer wavelength, that is the emission wavelength. This emitted light is often visible and can be of different color than absorbed light. The emitted light has a longer wavelength and therefore a lower photon energy than the absorbed radiations. Fluorophore. A fluorophore or fluorochrome, similar to a chromophore, is a fluorescent chemical compound that can re-emit light upon light excitation. When a fluorophore absorbs a photon of light at the excitation wavelength, it becomes excited to a higher energy state. It then quickly returns to its ground state by emitting a photon at emission wavelength. Fluorophores typically contain several combined aromatic groups or planar or cyclic molecules. Fluorophores can be categorized as molecules with intrinsic fluorescence and molecules used for extrinsic fluorescence. Now let us discuss both of these. Molecules with intrinsic fluorescence. These molecules naturally exhibit fluorescence without the need for external labeling or modification. These molecules have specific chemical or structural properties that allow them to absorb or emit light, resulting in their intrinsic fluorescence. The fluorescence properties of a fluorophore depend significantly on environmental factors, including solvent, pH, possible quenchers, neighboring groups, etc. Examples include aromatic hydrocarbons, aromatic compounds with conjugated pi-electron systems such as benzene, naphthalene, and anthracene can exhibit intrinsic fluorescence. The delocalized, that is free-moving pi-electron cloud in these molecules allow for absorption and emission of light. Flavins are a class of naturally occurring compounds, including riboflavin, that is vitamin B2, that exhibit intrinsic fluorescence. They are involved in various biological processes and are often used as cofactors in enzymes. Amino acids and proteins. Certain amino acids, such as tryptophan, tyrosine, and phenylalanine, can exhibit intrinsic fluorescence when they're part of a protein. This property is often utilized in protein studies and fluorescence spectroscopy. Chlorophyll, found in plants and photosynthetic organisms, exhibit intrinsic fluorescence when exposed to light. This property is essential for capturing light energy during photosynthesis. Nucleic acids, DNA and RNA can exhibit intrinsic fluorescence due to the presence of aromatic nucleotide bases like adenine and guanine. This property is used in DNA and RNA detection and analysis. Extrinsic fluorescence molecules. Frequently, molecules of interest for biochemical studies are non-fluorescent. In many of these cases, an external fluorophore can be introduced into the system by chemical coupling or non-covalent binding. Three criteria must be met by fluorophores in this context. Firstly, it must not affect the mechanistic properties of the system under investigation. Secondly, its fluorescence emission needs to be sensitive to environmental conditions in order to enable monitoring of the molecular events. And lastly, the fluorophore must be tightly bound at a unique location. Examples of extrinsic fluorescent labels include fluorescent dyes, small organic molecules with strong fluorescence properties such as fluorescein, rhodamine, and Texas red are often used as fluorescent dyes for labeling biomolecules like proteins, nucleic acids, and lipids. Fluorescent proteins, genetically encoded fluorescent proteins like green fluorescent protein, also known as GFP, are widely used to tag and visualize proteins in living cells and organisms. Quantum dots. Semiconductor nanocrystals known as quantum dots can be used as bright and photostable fluorescent labels for a variety of applications. Fluorescent antibodies. Antibodies labeled with fluorescent dyes are used for immunofluorescence assays to detect specific antigens in cells and tissues. Fluorescent nanoparticles. Nanoparticles functionalized with fluorescent dyes are employed in various research areas, including drug delivery and cellular imaging. Now let us see what is Stokes shift. It is the term used in fluorescence spectroscopy to describe the difference in wavelength or frequency between the absorption maximum, that is excitation, and the emission maximum fluorescence of a fluorescent molecule or a fluorophore. As you can see in this graph, the difference between the peak of absorbance and fluorescence is known as the Stokes shift. It is named after the Irish physicist George G. Stokes, who made significant contribution to the understanding of fluorescence in the 19th century. Now let us see various parameters for Stokes shift. Wavelength difference. The Stokes shift represents the shift of the fluorescence emission spectrum to a longer wavelength compared to the absorption or excitation spectrum. In other words, the emitted light is of lower energy and longer wavelength than the absorbed light. Energy dissipation. The Stokes shift occurs because during the brief time of a fluorophore is in its excited state after absorbing a photon, it can lose some of its energy through non-radiative processes like heat or vibrations. When the fluorophore returns to its ground state and emit a photon, this emitted photon has less energy and therefore a longer wavelength than the absorbed photon. Quantum yield. The extent of the Stokes shift can vary depending on the specific fluorophore and its chemical environment. It is influenced by factors like solvent polarity, temperature, and the presence of nearby molecules. A larger Stokes shift often indicates a lower quantum yield, meaning that a higher proportion of the absorbed energy is lost as non-radiative energy before fluorescence occurs. Importance in fluorescence spectroscopy. The Stokes shift is a crucial parameter in fluorescence spectroscopy. It helps differentiate between the excitation and emission signals, making it easier to detect the emitted fluorescence while minimizing interference from the excitation light. Application. In practical applications, the Stokes shift is important for the design of fluorescence experiments, including fluorescence microscopy, flow cytometry, and fluorescence-based assays. It allows researchers to choose appropriate filters and detectors to separate the excitation and emission wavelengths. Fluorescent labels. When choosing fluorophores or fluorescent labels for specific applications, researchers often consider the Stokes shift as it can affect the signal to noise ratio and the ability to distinguish fluorescence from background noise. Now let us see what is fluorescence lifetime. It is a fundamental property of fluorescent molecules or fluorophores that measures the average time a molecule spends in its excited state before returning to the ground state by emitting a photon. It is one of the key parameters in fluorescence spectroscopy and microscopy and provide valuable information about the fluorophore's environment and interactions. The fluorescence properties of a molecule are determined by properties of a molecule itself, that is internal factors, as well as environment of the protein, that is external factors. Fluorescence lifetime can be influenced by chemical environment of the fluorophore, changes in temperature, pH, solvent polarity, and the presence of nearby molecules such as quenchers or sensitizers can alter the fluorescence lifetime. The fluorescence intensity emitted by a molecule is dependent on the lifetime of the excited state. The transition from the excited to the ground state can be treated like a decay process of first order. That is, the number of molecules in the excited state decreases exponentially with time. The exponential coefficient, denoted by Kr, is called rate constant and is the reciprocal of the lifetime, as you can see here in the formula given. Now, before understanding the principle of fluorescence, it is important to know what are singlet and triplet states. These states refer to different configurations of electrons within molecules. These states play a crucial role in fluorescence and phosphorescence. Singlet ground state denoted by S naught. The singlet ground state is the lowest energy state of a molecule. In this state, all electrons are paired, as you can see here, meaning they have opposite spin orientations following the Pauli exclusion principle. Molecules in this singlet ground state are considered to be in their most stable and lowest energy configuration. Singlet excited state, denoted by S1, S2, etc. When a molecule absorbs energy, typically in the form of photon or light, electrons can be promoted to higher energy levels, creating what are known as singlet excited states, S1, S2, etc. In a singlet excited state, one or more electrons are in higher energy orbitals, which are still paired with opposite spins, as you can see here. Singlet excited states are relatively short-lived and are often associated with fluorescence. The emission of fluorescence occurs when electrons in a singlet excited state returns to the singlet ground state. Triplet excited state, denoted by T1, T2, etc., are higher energy electronic states that are corresponding singlet excited states. In a triplet excited state, two electrons have parallel spins, meaning they have same spin orientation. Transitions between singlet and triplet states are spin forbidden, which means they are less likely to occur due to the requirement of changing electron spin. Consequently, triplet states are typically longer lived than singlet excited states. Phosphorescence, which is a delayed emission of light, is often associated with transitions from triplet excited states to the singlet ground state. Now let us understand the principle of fluorescence spectroscopy. Fluorescence is an emission phenomena where an energy transition from a higher to a lower state is accompanied by radiation. Only molecules in their excited forms are able to emit fluorescence, thus they have to be brought into a state of higher energy prior to emission phenomena. A molecule in its electronic and vibrational ground state, denoted by S0 V0, as you can see here in the diagram, can absorb photons matching the energy difference of its various discrete states. The required photon energy has to be higher than that required to reach the vibrational ground state of the first electronic excited state, S1 V0. The excess energy is absorbed as vibrational energy and quickly dissipated as heat by collisions with solvent molecules. The molecules thus return to the vibrational ground state, S1 V0, as you can see here. These relaxations processes are non-radiating transitions from one energetic state to another with lower energy and are called internal conversions, denoted by IC. From the lowest level of the first electronic excited state, the molecules return to the ground state S0 either by emitting light, that is fluorescence, or by a non-radiative transition. Upon radiative transition, the molecules can end up in any of the vibrational states of the electronic ground state as per quantum mechanical rules. If the vibrational levels of the ground state overlap with those of the electronic excited state, the molecule will not emit fluorescence, but rather revert to ground state by non-radiative internal conversion. Now let us discuss what is phosphorescence. An associated phenomena in this context is phosphorescence, which arises from transition from a triplet state, that is T1, to the electronic singlet ground state. The molecule gets into the triplet state from an electronic excited singlet state by a process called intersystem crossing, as you can see here. The transition from singlet to triplet is quantum mechanically not allowed and thus only happens with low probability in certain molecules. As you can see here also in this previous diagram, intersystem crossing leads to the phenomena of phosphorescence. Such molecules usually contain heavy atoms. Phosphorescence happens with a long delay and persists even when the exciting energy is no longer applied. Phosphorescence is a photoluminescent phenomena in which a substance absorbs photons or light and then re-emits them at a longer wavelength with a delay after the excitation source is removed. These excited electrons return to lower energy state through a process called phosphorescent decay, emitting photons in the process. Unlike fluorescence, which involves the almost instantaneous emission of light upon photon absorption, phosphorescence involves a more extended time scale for photon emission. Now let us see the difference between fluorescence and phosphorescence. Parameter is time scale of emission. In fluorescence, the emission of light occurs almost immediately after the absorption of photon. The lifetime of the excited state is very short, typically on the order of nanoseconds. As a result, fluorescence appears as an instantaneous burst of light, whereas phosphorescence involves a delayed emission of light. After absorbing photons and transitioning to an excited state, the molecule remains in the excited state for an extended period before returning to the ground state by emitting light. This delay can range from microseconds to hours depending on the specific material. Phosphorescent materials continue to emit light even after the excitation source is removed. Mechanism of emission. Fluorescence involves transitions between electronic states in which electrons have opposite spin orientations, singlet-singlet transitions. These transitions are spin-allowed, meaning they occur more readily. When an excited electron returns to a lower energy state, it emits a photon. Phosphorescence involves transition between electronic states in which electrons have the same spin orientation, that is singlet-triplet transitions. These transitions are spin-forbidden, meaning they occur less readily. The spin-forbidden nature leads to the longer lifetime of the excited state, as the transition requires a change in electron spin. Phosphorescence typically occurs at lower temperatures than fluorescence because thermal vibrations can help overcome the spin-forbidden nature of the transitions. Common examples of fluorescence include fluorescent dyes, certain types of chemical compounds, and naturally occurring fluorescent molecules like green fluorescent protein. Phosphorescence is observed in materials such as glow-in-the-dark products, certain phosphorescent pigments, and some minerals like phosphorescent calcite. Energy considerations. Fluorescence often result in less energy loss because the excited state is short-lived and then return to the ground state very quickly. Whereas phosphorescence tends to result in greater energy loss because the excited state persists for a longer time before emitting a photon. Fluorescence is widely used in various applications, including fluorescence microscopy, flow cytometry, biomolecule labeling, medical diagnostics, and environmental monitoring. Phosphorescent materials are used in glow-in-the-dark products, emergency exit signs, and certain types of luminescent paints. Quantum yield, often denoted as phi, quantifies the efficiency of fluorescence in converting absorbed photon into emitted photons. Quantum yield is defined as the ratio of the number of photons emitted in a particular process, example fluorescence or phosphorescence, to the number of photons absorbed by the system. Quantum yield values typically range from 0 to 1, where a quantum yield of 0 indicates that none of the absorbed photon result in emission, representing a complete lack of efficiency, whereas a quantum yield of 1 indicates that every absorbed photon leads to emission, representing perfect efficiency. The quantum yield is a dimensionless quantity and, most importantly, the only absolute measure of fluorescence of a molecule. The effective lifetime of a fluorophore is dependent on all processes that causes relaxation, fluorescence emission, internal conversion, intersystem crossing, reaction of the excited state, quenching, fluorescence resonance energy transfer, also known as FRET. It equals the ratio of the rate constant for fluorescence emissions Kr and the sum of the rate constant for all the six processes mentioned above. Quenching. Quantum yield of a fluorophore is dependent on several internal and external factors. One of the external factors with practical implication is the presence of a quencher. A quencher molecule decreases the quantum yield of a fluorophore by non-radiating processes. The absorption process of the fluorophore is not altered by the presence of a quencher. However, the energy of the excited state is transferred onto the quenching molecules. Common quenchers include molecular oxygen, iodide ion, heavy metal ions like mercury, certain organic compounds, and various electron acceptors. Quenching rates can depend on temperature and the concentration of quencher molecules. Two kinds of quenching processes can be distinguished. First is dynamic quenching, which occurs by collision between the fluorophore in its excited state and the quencher. The energy from the excited molecule is transferred to the quencher, causing it to return to its ground state. And static quenching, whereby the quencher forms a complex with the fluorophore. The complex returns from the excited state to the ground state by non-radiating processes. Quenching studies can provide insights into the nature of molecular interactions and the mechanisms by which energy is transferred or dissipated. This information is valuable in understanding the behavior of molecules in various environments and under different conditions. Instruments used to measure the fluorescence intensity. First is fluorescence spectrophotometer, and then we have filter fluorometers. Both instruments are used to measure the intensity of fluorescence emitted by a sample. However, they differ in their mechanism of operation, flexibility, and the types of fluorescence measurements they can perform. Fluorescence spectrophotometer. It is a specialized laboratory instrument used to measure the fluorescence properties of substances. Light source. It typically includes one or more light source such as a xenon lamp or a high intensity light emitting diode, that is LED. These sources emit light at various wavelengths and user can select the appropriate excitation wavelength for the experiment. Monochromators or diffraction gratings or filters. Excitation monochromator. This optical component allows the user to select a specific excitation wavelength by adjusting the monochromator's setting. It ensures that only the desired wavelength of light reaches the sample. Emission monochromator. Similar to the excitation monochromator, it selects a specific range of wavelengths emitted by the sample for the detection. It helps isolate the fluorescence signal from other sources of light. Sample compartment. It is where the sample to be analyzed is placed. It typically includes a cuvette holder. Detector. A sensitive photodetector such as PMT or CCD is used to measure the intensity of emitted fluorescence. It converts the light signal into an electrical signal that can be processed by the instrument. Data acquisition and analysis. Modern fluorescence spectrophotometers are equipped with computer interfaces and software for data acquisition, analysis, and visualization. Researchers can record fluorescence spectra, measure fluorescence lifetimes, and perform other data analysis tasks. Temperature control. Some fluorescence spectrophotometers have temperature control system to maintain the sample at a specific temperature during experiments. This is important for studies involving temperature-dependent fluorescence changes. Now, since we have seen the different parts of fluorescence spectrophotometer, let's understand the working. Here, the excitation monochromator selects a specific excitation wavelength and the sample is irradiated with light at this wavelength. When the sample absorbs the excitation light, electrons are promoted to high energy states. As these electrons return to their ground states, they emit light at longer wavelength, which is detected by the emission monochromator. The detector measures the intensity of the emitted fluorescence at various wavelengths, creating a fluorescence spectrum. The obtained data can be analyzed to determine various properties, including the fluorescence intensity, emission wavelength, and quantum yield of the sample. Filter fluorometers. They operate based on the use of optical filters to select specific excitation and emission wavelengths. They are relatively simple instruments that can use fixed filters or filter wheels to isolate the desired wavelength of light. This simplicity makes them easy to use but limits their flexibility in terms of spectral selection. Whereas fluorescence spectrophotometers employ monochromators or diffraction gratings to scan a wide range of excitation and emission wavelengths. This allows for more precise control over the selection of excitation and emission wavelengths, making them versatile for various applications. Filter fluorometers have limited wavelength flexibility because they rely on fixed filters or limited number of filter choices. To change excitation or emission wavelengths, you typically need to replace filters or adjust a filter wheel. Filter fluorometers can be sensitive and are capable of detecting fluorescence signals, but their sensitivity may be lower than that of fluorescence spectrophotometers. Filter fluorometers are typically more cost-effective and budget-friendly than fluorescence spectrophotometers. They are a practical choice for routine fluorescence measurements with budget constraints. Filter fluorometers are suitable for routine fluorescence measurements such as quantifying known fluorescent compounds and conducting simple fluorescence assays. They are commonly used in clinical laboratories and for routine fluorescence analysis. Next slide shows a comparison between the fluorescence spectrophotometers versus filter fluorometers. Fluorescence fluorometers operate based on the principle of spectrophotometry. They use a monochromator or set of excitation and emission monochromatic filters to isolate specific wavelength of light. On the other hand, filter fluorometers use specific filters for both excitation and emission wavelengths. These filters are usually fixed and allow only certain wavelengths of light to pass through. Mechanism: a broad spectrum light source is used to excite the sample at a range of wavelengths. The emitted fluorescence is then dispersed into component wavelengths using a monochromator, allowing the measurement of fluorescence at different wavelengths. In filter fluorometers, the excitation filter allows only the desired wavelength of light to reach the sample for excitation. The emitted fluorescence passes through an emission filter, then transmits only the fluorescence emitted at a specific wavelength of interest. Advantages of fluorescence spectrophotometer: it allows for the measurement of fluorescence emission across a range of wavelengths, enabling the identification of multiple fluorophores in a sample, whereas in case of filter fluorometers, they are simple and easy to use, making them suitable for routine fluorescence measurements, cost-effective and suitable for applications where detailed spectral information is not necessary. Fluorescence spectrophotometer offers flexibility in selecting excitation and emission wavelengths across a broad spectrum, whereas filter fluorometers give limited flexibility. Fluorescence spectrophotometers are commonly used in research applications where detailed spectral analysis is required, such as molecular biology, biochemistry, and environmental science, whereas filter fluorometers are more suitable for routine measurements in applications like clinical diagnostic, quality control, and simple fluorescence assays. Applications of fluorescence spectroscopy: biochemical and medical research, protein and nucleic acid analysis. Fluorescence spectroscopy is used to monitor protein folding, ligand binding, and DNA hybridization. Fluorescent labels: fluorescent dyes and probes are used to label specific biomolecules for tracking and quantifying cellular processes. This is essential in cell biology, immunology, and molecular biology. Clinical diagnostics: fluorescence-based assays are used for clinical diagnosis, including the detection of biomarkers, pathogens, and diseases such as cancer. Environmental analysis, water quality monitoring. Fluorescence spectroscopy is used to assess water quality by detecting contaminants and pollutants in natural waters and wastewaters. Environmental chemistry. Researchers use fluorescence to study chemical reactions, trace pollutants, and environmental processes in air, soil, and water. Chemical analysis. It is used for quantitative analysis of various analytes, including ions, metal complexes, and organic compounds. Food and beverage industry. Fluorescence spectroscopy assists in food quality control and safety by detecting contaminants, pathogens, and adulterants. Food chemistry. It is used to study flavor compounds, antioxidants, and other components in food products. In pharmaceutical industry, drug discovery, fluorescence spectroscopy is used in high throughput screening to identify potential drug compounds that can interact with specific antigens. Pharmacology, it helps study drug receptor interactions and drug metabolism. Forensics. Forensic science, fluorescence spectroscopy aids forensic scientists in identifying and analyzing trace evidence such as blood stains, fibers, and drugs.